Welcome everybody to our physics colloquium series. We're delighted to have Professor Margaret Gardel here today to talk about biophysics, and I will let Christoph Schmidt do a more formal introduction. Before we do that, I just want to let you know that Professor Gardel, her preference would be for people to go ahead and interrupt with questions during the talk. The more discussion, the better, I think. And so I will monitor the chat and raised hands, and just let us know if you want to ask a question. Our next colloquium will be March 31st by Xiaoming Mao from the University of Michigan, who will be talking about topological mechanical materials. So we have a couple of talks on mechanics of one sort or another in a row here. So I'm looking forward to hearing the presentation today, and I'll turn it over to Christoph to introduce our speaker.
Thanks, Josh. And yeah, thanks, Margaret, for accepting our invitation in this busy time, being involved in the APS organization of DBio, I guess, all day long. It was great that you could still make the time, and unfortunately not in person, but it's great that you're giving us a colloquium. Margaret is a professor of physics and also at the James Franck Institute and the Institute for Biophysical Dynamics at the University of Chicago, and is also the director of the Chicago MRSEC NSF Center. She did her PhD in Harvard in Dave Waits's group, and that's when I first met her, when she was working on rheology and soft materials and actin networks in Dave's lab. This was in the early 2000s, and she then went on to postdoc first at MIT. She actually was a Pappalardo Fellow at MIT, which is a very prestigious fellowship, fairly independent. And this was also more on the side of soft matter with some aspect of bio, but then she really dipped into cell biology, doing a postdoc with Claire Waterman at the Scripps Institute in San Diego. And I think since then she has strong credentials in the biological side also of soft matter physics. Since 2007, she has been at the University of Chicago and has built a very impressive operation, and is doing a lot of biophysics, cell mechanics, and we have a lot of overlapping interests, and there's very interesting work coming from her group, in her lab, and her surroundings. She's a fellow of the American Physical Society. She got an NIH Director's Pioneer Award and many other fellowships and awards that would take too long to all name here. So thanks again, Margaret, for coming, and I look forward to your talk.
Thank you very much, Christoph. Yeah, it's fun being able to see you and Brent in the same day. It's like two of my oldest buddies. And thank you all. Okay, so I've tried to design this talk for as broad of a physics audience as I can. And I would really like it if people stop to ask me questions if it gets too jargony at a point in time. And so broadly, I'm interested in understanding how cells and organisms control and change their shape. So these two movies here are phase contrast movies of a single endothelial cell crawling through a fibrillar matrix. It's actually from a mouse aorta, and the single cell escaped the aortic tissue and is crawling through a collagen matrix. And this one on the right is the first 24 hours of a zebrafish embryo, where it starts out as a single cell, but over the course of a day, you can see the shape and body form of a fish form. And so where the scale at which I think about these questions is that between these 4 orders of magnitude between that of single molecules, which form the basis of materials that are the physical constraints of how shape and forces are generated at the cell scale. And this has been a really exciting time to be at this sort of length scale, because molecular cell biology is now firmly established as a powerful tool in cell biology. There's lots of ways to understand the structure and function of single molecules, and to modify the molecular composition of living tissue. And so we can think of this general field as materials and machines that are built out of proteins, which have their own enzymatic activity. They are molecular machines. They take chemical energy and undergo conformational changes that can generate forces at the molecular scale. And we need to understand how those forces then are transmitted through this material to be able to drive these beautiful behaviors. And then, so this is, you know, I missed a word here. It's either this biological active matter or living matter is sort of the class of materials, you know, condensed matter physics that we think of, right? So it's a sort of a subset of soft materials where we think about active processes within materials. In my lab, we focus this a little bit more to think, when you think about cell shape control, really the actin cytoskeleton is what's controlling the shape and motion of adherent cells. So this is a fluorescence image where the stain, the signal here is coming from staining against a filamentous protein termed actin or actin filaments. These are semi-flexible polymers. So you can see that there's places in the cell where there's fine meshworks or thick bundles. And there's been much progress made on characterizing the different kinds of machines made out of these actin-based networks. So at the front of the cell, there's actually protrusive forces generated by a dense branched network of polymers that are growing at one end, and there's polarization of the polymers so that you generate a protrusive force. And then throughout the body of the cell, there's contractile tension generated by a collection of actin filaments, and then another molecular motor, myosin 2, that translocates actin filaments. And then, of course, you have pushing forces and contractile forces, and the way these get transmitted between the cells and their environment is through adhesions. So there's other complexes that assemble and form connections between neighboring cells or between cells and their extracellular matrix. And it's this coupling between this protrusive and contractile and adhesive machinery that allows cells to control their shapes. And so each one of these organelles, we have fairly good understanding about molecules that associate with these organelles, but there's still a lot left to understand the physics of these materials, okay? And so for the first, I would say, you know, really 11 years of my group, I was really interested in the active component of these materials. And if I summarize this efforts, not just by my lab, but the whole field, and it's this question of how do you take a molecular motor, an enzyme. So this is a schematic, a picture of a half of a myosin 2 filament that then translocate this actin filament on top. How do you take these molecular scale enzymatic activities to generate materials that are contractile or generate stresses at cellular scales? So how do you span from molecular scale biophysics to living materials to understand cell and tissue morphogenesis? And you can think about these as two different types of scales of questions of how do you generate forces from actomyosin sliding. That's one class of questions we were asking for a while. But then how do you describe materials with distributed active stresses? So how do you make descriptions of elastic solids with active stresses or viscous fluids with active stresses? And that's more of a continuum scale. So these two ways have really helped us bridge these highly disparate length scales. As we and the field have gotten better at these approaches, I would say that something that's really a conundrum for me in the past couple of years is the fact that we know that biological materials, the parameters, the order parameters that describe them, say their elasticity or their viscosity, are not constant in time. And biological materials, these parameters actually change. So a cell might change its continuum elastic parameter as it's moving around or it's changing shape. And so how do we describe a material what are the rules in which these parameters are adaptive and changing over time? Or there might be spatial heterogeneity in these mechanical properties. So in an epithelial tissue, you might have different spring constants or elasticities at different junctions. What's the origin of these heterogeneities and how do we describe them? A real big challenge I think now going is how do we describe adaptive and active materials? So one way we sort of been classifying it now, and I'll describe one particular problem we're thinking about, is that now we need to not only have the active matter part of this biological materials, but also how these materials are sensing forces at the molecular scale, and then how these force sensitive processes work together to build an organelle, something that's at the subcellular scale that is that modifies its mechanical properties in response to applied forces. And if we understand this, in addition to the activity parts, then we can start to understand morphogenesis, like have predictive models to describe cell motion and cell shape change. Okay, so of course we study one very small projects in this sort of bigger vision. I'm going to describe to you today. Part of my talk is going to describe a morphogenetic ratchet, where basically the adhesion between two cells and how length changes occur. I'm not going to be able to talk to you about a force sensing mechanism, which we recently published earlier last year, just because in terms of fitting everything into one talk, it didn't make logical sense. So I'm really going to be focusing on these morphogenetic behaviors of epithelial tissue. Okay. So where we're going to be situated today is at the scale of epithelial tissue. Sorry for the spelling error. And these are two of my favorite movies. One is a beautiful image from Matt Tiska at Vanderbilt of an intestinal organoid. So this is a formed from intestinal stem cells. They've made this organoid, and now the different colors are different height positions. And so as the intestinal organoid is changing shape, you can see, and the fluorescence here is just a marker of what the cell shape is. So as the organoid is changing shape, you can see the shape of cells are also changing. The connections between cells are lengthening and shortening as cell division occurs. On the right is one of the experiments I'm going to describe to you from my lab, where we've made a model epithelial tissue by taking a cell culture, tissue culture cell, and proliferating it. This is formed of MBCK cells, a canine kidney cell. And the marker here again is of the cell perimeter, and you can see these popping events where cells are dividing, and hopefully as the movie loops, you also see the cell shapes are rearranging. And so the question is, how does the shapes of cells in these epithelial tissues get determined? And on the right, we're also going to start asking these questions of, like, at what point does cell division get arrested? How does proliferation then become arrested in an epithelial monolayer? Okay, so if we want to think about, I've been showing you movies of the cell boundary. If we want to understand how shape is regulating epithelial tissue, we have to think back to the actin cytoskeleton. So at the boundary of each cell, now this is staining for that polymer actin, and we look at the top surface of these cells, there is a belt of actin around the boundary of the cell. And this belt, apical belt of actin and myosin, is what's controlling the shape of the cells on that layer. On the bottom surface of the cells are where it's connecting to the extracellular matrix. In addition to the actin, so this is now just immunofluorescence for actin, but also an adhesion molecule that's connecting the two cells together, but also these myosin 2 motors that are generating contractile tension. So it's this actin-myosin bundle at the junction that is determining the forces that are exerted on each junction. Okay, so traditionally, now almost over 100 years ago, when developmental biologist and mathematician Sir Darcy Thompson looked at this epithelial tissue, right, he was reminded of soap bubbles, like compressed soap bubbles, right? And there's lots of similarities, right? The shape of these soap bubbles is really determined by the compression and the large amount of crowding from the neighboring soap bubbles. And this led to the development of vertex models of tissue mechanics that are still widely used today. And in here, the energy of the energies are really determined by a spring constant that of the compressibility of the cells, the area compression of the cells, and also the sum of all the line tensions. So the contractile force of along each of the junctions. And so by minimizing this energy, you can then make predictions of the shape distributions in the epithelial tissue. Now, this vertex model is an elastic model. There's no dynamics in it. So it's most useful when you just take a snapshot and you look for distributions of shapes. And so this is, I sometimes talk to biologists. So definition of elasticity in this case is that it's reversible. So if you apply a force, the length, increase the force on the junction, the length might shrink for a bit. But if you take off that force, the length recovers, right? There's no plasticity and no length changes. Now, remember those initial movies I showed you, there is motion and dynamics. So the way you add in active stresses into is to think about the center of mass of each of these tissues as having a spontaneous a force that distorts this entire lattice. So these active stresses then drive the motion of the epithelial tissue to make what is known as an active vertex model. Okay, so what I'm going to tell you today is I'm going to tell two different scales of our work in this area. One is a modification of this elastic-like description of junction mechanics, where we find an adaptation or plasticity of how junction length changes over time when it is experiencing force fluctuations. And the other one, which I'll tell first, is about how to describe the active stresses and what those active stresses are determined, how those active stresses are determined in the epithelial tissue.
Can I ask a quick question?
Yeah, of course, please.
So I think epithelial means related to skins or surfaces or something, right? So I'm sort of curious, what's the, how general is this? If you study this, is this something that's true of all organisms, or are you studying this in a fruit fly and it's specific to fruit flies, or?
Yeah, there's lots of different examples of, so epithelial tissues are all flat. Simple epithelial are one layer of cells that form like a two-dimensional, a plane. And there are lots of examples of that in developmental systems, like in fruit flies, in C. elegans, right? In humans, right? Skin is an example of a stratified epithelium that's multi-layered. So I would say that the strengths of the approaches we use, we don't study this in a certain developmental context. What we do is try to build systems in which we can then do enough perturbations to actually break down physical models. Does that make sense? So like I live in the space where we want to do enough biophysics that we can really kill the models.
Great, thank you.
Okay, there's lots of great epithelial examples in development, and I'm always jealous of those systems. But there's lots of other forces going on. So when we build model epithelial from tissue culture cells, we have lots of control over what those epithelial tissues are like. Okay. Okay, so these are going to be, there's two parts, and there's just going to be at different scales. So the first question is this question of active stresses. Where are these active stresses coming from? Okay, so this is now a description of how this experiment I showed you, this movie I showed you was done. This work is now published in PNAS. Shows you how long things take to publish. We initially posted in bioRxiv a year and a half ago or something like that. But it was just published in PNAS about 2 weeks ago. So what John Devaney did, and he really drove this entire project, a really talented grad student, he puts a soft collagen gel down and then seeds these MDCK cells on them. Initially, these cells are sparse enough, so there's gaps. So that's what you see here in the black. At a certain time point, which we identify as T equals 0 hours, those cells form a confluent monolayer. And then over time, that confluent monolayer remodels to time, which is around 12 hours later, where there's no more dynamics, okay? So we're really defining t equals 0 at the time point when you form a two-dimensional sheet with no holes in it. And anytime you see a black spot in the movies, it's because there's a little portion of cells that aren't expressing the protein he's using to visualize the cell shape. Okay, so this is where I think the methods that Daniel and Christoph's group might be helpful. This is now using old segmentation to track the identify cells in this epithelial tissue and identify the speed and the shape of these cells. So as you watch this process happening, so maybe focus on the left here, you can see that initially there's lots of motion right now, and then the motion slows down to the point where all the motion is really arrested. And so the cell speed is decreasing over this time, and the cell shape is also evolving over this time. John takes the average of the cell speed and the cell shape to then and plots them as a function of time. So this is the speed, the average cell speed. You can see initially it's a high speed. It then becomes arrested, where it doesn't change over time. And the question is, like, what's causing this arrest, right? That's one question. It's like, why did the cell stop moving? And there's a few. Yeah.
To clarify, are cells dividing also, or
they are? Yeah.
So
yeah. So you can and this can come up again because it's actually something that we're just now appreciating. Yeah. So you see lots of division going on here. Tons and tons of division.
So what is conserved? Is the volume of the whole tissue conserved, or are they growing at the same time? Or?
Yeah, that's a great question. I'll just give you the answer. We are in a phase where the cell volume is decreasing. The individual cell volume, this volume of the tissue is conserved. That's what we think of it as. And the cell volume, when they're undergoing divisions, is decreasing.
But they do get taller, right? The Z
they
direction?
do not get taller.
Oh, okay, good.
No, it's actually really, this is, I mean, for people, I mean, I'll only speak to Christoph and Josh because you asked a question. So Boris Schraman, of all people, has this paper from about 10 years ago that defines these epithelial tissues at different stages of density. And at a very low density, they actually proliferate and they go back to their original, the cells go through, proliferate and then go through their same volume again. But when the tissue gets sufficiently dense, then the cell volume, and when they go through a division, they just divide and they don't actually grow anymore. And we're in that second regime, but it's a really cool, they grow a little bit, but it's another thing where that growth rate is really changing as a function of mechanical stress, I think. Yeah. Anyways, I could go on about that because we did not realize this until about 3 months ago.
So then I just have to ask,
yeah,
what's the boundary condition? Are these?
Yeah, so I mean, the thing is that it's on a gel that's in like a sample chamber, and so there is a boundary. I think the effective boundary is that you're in this big amount of tissue where the cells are coupled to a gel. Yeah, there is a constraint on the side, but I just think that the cells don't want to push each other. There's just too much mass around them.
So is it just my impression, but the mean free path between cell motion is decreasing with time as well?
Path. So like the mean square displacement of each cell, like
well, the mean distance that the cell moves before it interacts with another cell and feels stress from it.
Yeah, so that is that's another way of characterizing the speed, right? Like this is an instantaneous speed, but if you actually calculated like a mean square displacement, you would also see that decrease.
Okay.
Yeah, so it becomes more cage-like, you know, at those later times.
So then
those things are really hard to characterize because you know, statistics on mean square displacements. I don't. Anyways, yeah, it's harder to get those long lag times.
So can I think about how they're interacting globally as the stress is part of the global feedback for the system? Like
the whole tissue stress.
Yeah, if you add it up as a collective system.
Yeah. If sort of, yeah, I would have said it slightly different, but yeah, I think sort of, because I think the stress is really then determined by the mechanics of each cells, and that's what we think that really at some point when Mike, can you ask that question after? I'm not sure if I understand fully your question. I'm not sure if I'm trying to
It
think
may
about
not have been a good question, so yes.
No, I
thank
just, I think stress is playing a role. Whether it's playing a role in the dynamics or the proliferation is something that I don't have a concise answer to at this moment.
Margaret, do you have ability to stretch or compress the substrate to impose global deformation to slow down or speed up the motion?
Yeah, we do not on this system, but that's definitely feasible with this kind. I mean, that other people do. Yeah, that would be a great thing to be able to do. I mean, people have definitely done it in this type of model. It's a really good, it'd be a good thing to do. Okay, so let me try to retract to where we were. So the speed is going down, the cells stop moving, and as was noted that the cells are also proliferating, and the question is, are they continually proliferating? And if you measure the density of the cells, the density also is increasing, so the proliferation rate is high, and then the proliferation rate decreases. So the density also increases and then sort of stabilizes at those long time scales. So the cell division is the proliferation is stopped, and the motion is also stopped. Okay, so this is one model that where the motion of the cells is controlled by the density. All right. Another model that would be is that the cell motion is then controlled by a cell shape, and I'll get to it in the next thing. But the cell shape is sort of a marker of mechanics of the tissue. And so here, the way we parameterize cell shape is known as a shape parameter, which is just the ratio of the perimeter of the cell to its square root of area. This is a dimensionless number. So as the cells start out with a more elongated shape, and as time goes on, they become more circular or more hexagonal, really. And so this, both the density is going up and plateauing, but the shape parameter is also stabilizing, so cells are becoming round. All these things are happening at the same time. Both models that predict arrested motion because of cell density and shape have both been predicted. And the first thing we want to do is just test to see whether we could distinguish between those two different models. Okay. So with this, we collaborated with Lisa Manning and Daniel Sussman, who was her postdoc at the time, but now has started his own position at Emory. And Lisa has really been one of the leaders in establishing developing active vertex models to make predictions about how the motion of epithelial tissue. And to summarize, sort of briefly summarize what this model is saying is that by different types of approaches, you can characterize an epithelial tissue by the shape parameter, that's that shape of the cells, the average shape, and think about it as having an active stress. And so if the shape is parameter is very high, then over a large range of active stresses, this tissue will be very fluid-like. There'll be lots of neighbor rearrangements and remodeling that will lead to a tissue that is effectively fluid-like. If you go through a boundary to say decrease the shape parameter or decrease the active stresses, now there's fewer rearrangements and the tissue becomes effectively solid-like. So our first question was our experimental data consistent with this picture of a fluid to solid transition, and would it predict at the expected shape parameter? Okay, so Daniel was able to perform simulations of the self-propelled Voronoi model. And what he did was consider a range of active stresses and shape parameters. So these are his parameters in the model. And he took different paths of going from point A to point B, right? So these are different paths he took. And with all of these in the model prediction that now we plot the mean square displacement, but that you could think of as the speed, is that the instantaneous speed as a function of shape parameter, they all should arrest. The speed should go down at a shape parameter of around 3.8. All right, so this predicted that the dynamics within the tissue should all become arrested at a singular shape parameter that is pretty independent of the path you take to get there. So when we plot our experimental data on this, now we're taking the same data I showed you before. And so time goes to the bottom left. And we see that it really evolves over time in a path that looks vaguely similar to this model. And at a point, which is 3.87, which is not that much different than what's predicted, which is pretty similar to what's predicted from the model. Okay, so this is one experimental data. It's consistent with this theoretical prediction that the arrest is coming about when there's at a certain shape parameter. Now, this type of prediction had been made before, namely in other types of experimental systems in development, but also in other types of tissue culture cells, but there hadn't been a lot of perturbations to the systems to be able to see if this was always the case, how universal this was. Sorry, this is just a picture of what the cells look like at the initial time. There, see how they're more elongated to the final time when they have that more hexagonal shape. Okay, so what John did then was basically do a whole slew of experiments where he could do these same types of remodeling experiments on different matrix stiffnesses and different perturbations to the epithelium to see at what point this model predictions still hold or not. So this is the best example we have to test it. It happened to be an experiment where he put the cells on different stiffness collagen matrices. And so on the right here, you see the speed of the cells as a function of the number density. So you can see that this really affects the dynamics in terms of how what the speed is at different densities. So for softer tissues, the cells can become much more dense before they arrest their motion, whereas at the stiffer matrices, their motion is arrested at much lower densities. But when we plotted this speed as a function of the shape parameter, now you can see all this data basically collapse onto a similar line, and they all become arrested at a similar shape parameter, which really suggests that it's the shape parameter and not density that is the important parameter in terms of controlling the speed. Okay, so that was great. But then he started doing other experiments. And I'm not going to tell you exactly what all these perturbations do. These change how well the cells protrude or contract. And he was able to get all other experiments where he had the cells arrest at different shape parameters. So now this becomes the problem, and we had a big fight with Lisa and Daniel about how to explain this data, right? Because this data was not consistent with the theory of this jamming transition. And now, of course, you plot the final shape parameter as a function of, say, the density, and you see it's just scattershot, right? There's no rhyme or reason to any of these perturbations. Okay, so at this point, I don't actually know what led him. I think he sort of had some hunch that he noticed with all these perturbations that what seemed to be changing is how fast the cells were proliferating. So then what John did was extract out a measure of cell proliferation rate, like what fraction of cells were undergoing division at any given time, a rate, and then plot that cell division fraction and plotted the shape parameter as a function of the cell division fraction, and saw this really nice collapse of the data. So this whatever final shape parameter you get, it's really dependent on how fast the proliferation of the cells is. And even you could take another type of epithelial tissue, a Keiko2 cell line, and it even fell on this data, right? So this was really exciting. And so then we looked in the literature, and there's been lots of models. What this data really suggested to us is, and we weren't really quite convinced yet, is that cell divisions or cell cycle regulation, cells going through the cell cycle, are really a source of active stress in the tissue. That maybe if you change how many cells are going through the cell cycle, that's going to affect how much active stress is. So this has been proposed before. This is not a new idea. There's one hypothesis out there that the stresses in an epithelial tissue from cell division come from like an active stress dipole that's coming because cells deform when they're undergoing cell division. And so it's actually a source of a contractile dipole. John looked at this and looked at the distortion field around the cells, and there isn't a large or long range distortion field, so he ruled that out. It's also been proposed that cell divisions could change the shape of the cells because there's oriented cell divisions. Cells tend to divide along their long axis. John looked at this and then decided that this oriented cell division effect could not account for all the changes in cell shape that he was observing. So we ruled that out. And then we found this one paper from a few years ago. And there's two other papers in this with this idea is that just the cell cycle regulators actually change the mechanics of the adhesive and the contractile machinery of cells. so that there's really a cell cycle dependent changes in the cortical tension and adhesion in the epithelial tissue. And this is something now from a cell biological perspective we're now looking into, because as far as I can tell, this is the only cell biology paper on this topic. So there's a lot to be understood in this. Okay, so this is the cool experiment. So, and this is for the students, the audience, like your advisors love it when you do an experiment that you didn't tell them to do and totally stresses them out because they don't know anything about the cell cycle. So this is the same experiment, the remodeling experiment. And what John's going to do is add the CDK1 inhibitor. But this arrests all the cells in the cell cycle and prevents cells from going through the cell cycle and dividing. Okay. And then he's going to wash it out. And so here the cells are moving around like mad. He washes in the inhibitor. The cells stop moving immediately. Then he washes it out and they start moving again. And now the movie loops again. Here the movie's starting. You push in the inhibitor, everything stops and freezes up. You wash it out and the cells move again. So basically by stopping the cell cycle, all the motion in the epithelial tissue gets immediately arrested. Everything stops. And then when you wash it out, everything starts running again. So this really suggested that the cell cycle, and for those of you who care about Rho and ROC and myosin activity, like all those perturbations did nothing really. Like we're completely redundant. The CDK1 really was the most critical perturbation that we found. This is a quantification of that data, and the pertinent graph here is the speed. So before the inhibitor, the cell speed is decreasing. You wash in the inhibitor, it just stops, it goes down. You wash it out, the speed increases again, and then it decays again. Okay. All right. Okay. So now how do we put this into Lisa and Daniel's model? Well, this suggested to us that during the cell cycle, there is a cell cycle dependent changes in the junction mechanics. Okay. And so perhaps on the right hand, that schematic that I drew is the most useful. So what we're thinking is that in this epithelial tissue, the cell cycle is not synchronized in the epithelial tissue. And what this means is that if we think about the tension, that contractile tension on the junctions, there's a very broad distribution of the junctional tension. Once the cell cycle is arrested, say everything is stuck in this G1 phase of the cell cycle, there's a much narrower distribution of the tension on the cell cycles, on the junctional tension. So now what Daniel can do is and this was really done by, and it happened that another previous postdoc in Lisa's group, Takaki, Takaki is actually his first name, so this is a wrong citation, had recently published a model where he considered what would happen to the active stresses in an epithelial tissue with a distribution of junctional tensions. And so these are two snapshots of his simulation results where there's a low variance in the junctional tension, and you can see the motion is very small. This is like a path of the centroid of the cell over time. Or on the right, a high variance in the junctional tension. You can see these cells are moving a lot. And so now by just changing the distribution, tightening it or widening it of the tension on the cells in epithelia, you can modify the active stresses in there. Okay. And this was super exciting. So this is the picture that we had. This was our understanding. This is the understanding in the paper that we just published. I will say, like the comment that was made before, is that we now appreciate that when the cells are undergoing cell division, they aren't generating active stress simply because their volume is not really changing sizably. They're undergoing a cell cycle without really cell proliferation, which is usually controlled by mTOR signaling. And so now we think of this as a really interesting system to think about why the cell cycle, first of all, why is the cell cycle changing junctional tension? What's the mechanism of that? But also, the cells stop dividing. The cell cycle becomes arrested naturally because of crowding in the epithelial tissue. So there's a mechanical feedback between the cell mechanics of the epithelial tissue and control of the cell cycle signaling. And this really gets to this really fundamental question in tissue mechanics of how cells regulate their growth and proliferation and in a crowded epithelium, which is something we're really excited about looking at in the future. It's getting into areas of biology that really I don't know anything about, so I'm trying to learn those because I've tried to stay pretty focused to the actin cytoskeleton in my life. Okay, are there any questions on that section? Because I'm going to switch gears a little bit now. Okay. Good. Okay. All right. So if you fell asleep, now's a good time to wake up again. Okay. So the second half of the talk is I'm going to highlight some results from a really wonderful collaboration we had with Sheila Ditcher Banerjee, who's now at Carnegie Mellon. He moved from UCL about a year ago, and his graduate student Mike Stadden, and a graduate student of mine, Kate Kavanaugh, who just graduated, we just posted a third paper for this collaboration on bioRxiv, which I won't have a chance to go into depth about. And this is this project is really focused on the single sort of the mechanism at the scale of a single junction, a single cell-cell interface of how its length is controlled. And we were really inspired from work in the developmental biology field, where they described junction, that's what they call the cell-cell junction, length changes as a morphogenetic ratchet. And so what do we mean by that? Well, what we mean is that there's pulses of assembly of this actin-myosin contractile machinery that drive shortening of the junction. And when this pulse of force happens, the junction length decreases and it's irreversible. So that after that pulse of that contractile force is removed, the length is now shortened. Okay, so it's just acting like a ratchet. And this is controlled upstream by Rho GTPase signaling. So this has been described for been observed for quite some time now in multicellular tissue. And this is some citations that have been seen over the past 10 years. It turns out, and this is really cool because, and something that we're trying to figure out how to study more sort of coherently, when you think about the materials inside cells, it's not like you assemble them and they're static. They're constantly assembling and disassembling, and they're controlled upstream by these biochemical factors, right? So some people like to think about these things as an excitable system, right? Where you have these chemical signals that are controlling these assembly of materials. And so that pulsatile row is an example of such an excitation. This pulsatile row has also been observed in adherent cells. So this is an example of row pulses in just a spread fibroblasts. And more recently, there's been a few really nice pieces of work about describing the cell cortex as an excitable system of this feedback between chemical and mechanical signaling. So we were wondering if we could really study this process of a ratchet at the single junction level by using a new tool that's become available called optogenetics. Okay, so to get there, I just need to give a brief discussion on what we're going to be controlling here. So to control the contractile machinery of actin and myosin requires activity of this Rho GTPase, which then assembles actin polymers and myosin 2 filaments to then generate contractile force. And this Rho GTPase is controlled by cycling of these guanine exchange factors and gaps, which all seem to predominantly occur at the cell membrane. Okay, so we want to turn on this Rho GTPase to assemble the actin-myosin. Okay, so what we want to do is we're using this tool that was developed by a colleague of mine, Michael Glotzer, called Tulip. And what this is a system that was adapted from a photosensitive interaction. And what it consists of is this love peptide, which is a love domain with this J alpha helix and this peptide. So in the presence of blue light, this J alpha helix of the love peptide unfolds and exposes a high affinity binding site for this PDZ domain. So in the presence of blue light, you have a high affinity binding partner between these two tags. And then if you turn off the light, they are not bound to each other. So when you shine blue light, these proteins come together and stick. You turn off the light, it's reversible. Okay. So what Michael did was then attach this PDZ handle to something that turns on and off that row, right? That row signaling to assemble actomyosin and attach the LOVE peptide to the membrane. So now whenever you shine light on the cell, this Rho activator will immediately go to the cell membrane. And so this is an example of how it works. You shine light right there. This is where the tag is localized. It goes to the place where you shine the light. I guess I don't show the light there, but that's where the light is going. And the kinetics are very fast and reversible. So when you shine light, everything accumulates, and then it dissociates back to its pre-activation levels. Okay, so this is fun because, and this is probably one of the most fun, it's really hard experiments, but really fun because you can yank cells to do what you want them to do at any given time. So here's an example where Kate shined light right on a junction, and then you can see the junction contract. So we can tell the junction to contract when and where you want it to go. And so she can just measure the length of the junction over time. And you can see for this experiment, the junction contracted, and then you turn off the light and it goes back to its original length. So that's not that surprising. This is very consistent with the vertex model, right? Nothing surprising there. But with this tool, now we can ask what happens if we change the intensity of the light? What happens if we change the duration, right? All these great tools for dynamical systems that we can now do inside living cells. Okay, so now when she shines the light on it for longer, she sees irreversible shortening. I guess you could probably guess that from my buildup. And so when she shines light and doubles the duration, now the junction contracts and then stays contracted even after she turns off the light. And this is where where it can get fun is now you can quantify what the duration is of the activation that requires this irreversible shortening. So if you plot the final length of the junction as a function of activation time, you can see for very short activations, in this scale 5 minutes, everything is reversible. And then as you increase the activation time, everything shortens, but you can see it's not linear. It sort of goes down to another plateau. So this is a cool sort of design principle, right? It sort of buffers the system to very short-lived signals, right? It says, oh, if you get a short-lived signal that wants you to contract, it's just going to not remodel. But now if the signal gets longer, it's going to remodel, okay? Likewise, for amplitude or long durations, it's not going to contract, it's not going to shorten uncontrollably. It's just going to go down to a new set point. And then you need a new pulse to be able to get a further cue. And so this irreversible shortening required modifications of the vertex model. And that's what Sheila and Mike did. They tried there's lots of variants of the vertex model that have been used to account for different types of remodeling of junction length. And most of the published ones all the published ones did not work. So they had to come up with their own rules, which then we could test with our experiments. So the rule that they found worked was that if we now plot the say the rest length remodeling of the junction, that's just the length of the junction as a function of time, as it's either extended or compressed, it has a constant remodeling. There's an increased remodeling rate as a function of strain. And then there's also a remodeling of the junctional tension. So how much contractile tension is in the junction. And there's no remodeling at low strains, but at a certain critical threshold, that junctional tension remodels. So it's the really critical thing is this strain thresholded tension remodeling. And with those features, the simulation results are able to recapitulate what we see experimentally. And sort of to get the essence of what is physically going on here, I just want to alert you to this bottom graph here. So you can see junction strain, that's how much this junction is being compressed or extended. What happens is when you first shine on the light, when there's an acute part of contraction, the strain momentarily goes above this threshold for remodeling, and there's an acute remodeling period. But as the strain and as the length and the tension remodel, that gets below that that threshold, so there's no subsequent tension remodeling. So all that tension remodeling is happening acutely in those initial periods, but takes enough time. So that's where you require enough time to be able to have tension remodeling. Okay, so then we could go through and test all the kinetics. So this is what makes these optogenetics is really allowing us to test all these mechanical models of living cells in a way that I've never thought was possible. We can now test the kinetics of the model, the parameters in the model to confirm that the experiment matched the model. And we can also change the amplitude of the light sensitivity by changing the input signal and show that that also was consistent with the model. This is super exciting way to be able to approach cell and tissue mechanics. And so then I'll finish with this one thing. The other types of things you can start to now ask what the spatiotemporal structure of these pulses, what the consequences of those are. And specifically, you can ask what's the difference between one pulse of long duration and two pulses of short duration. Now, for this audience, given that I already gave you some insight to how the remodeling occurs, I think you probably can guess the result that these are going to be quite different. So what happens when with the one pulse, what happens is you do all that remodeling in that first initial period, and then everything saturates out and you only get to a single plateau. That's not any different. I showed you that saturation. If you have this separated by a certain amount of time, now you can actually proceed through two subsequent remodeling phases so you can get to a shorter junction. All right. So now you can do this plot. This is simulation results of what the final the contracted length is as a function of the total activation time and the rest time. So this is the time between pulses. And let's just take a time here of 60. So now you can see is that if your rest time is very short, you're really limited by that. It looks like a single pulse. It doesn't really make, you can't do any further remodeling, but as you increase the rest time, then you can really overcome this to get to that second pulse of contraction. So it speaks to this idea that if everything is ratcheted, it gives you an idea of what the kinetics are to be able to control that ratcheting behavior. Okay, and experimentally, we can go in and test this idea where we do two different pulses and see the lower junction length. Okay. I'm going to have one slide on what we think is happening here, like cell biologically. So when the contraction is occurring here, you are compressing the whole junction. And what we think is when that compression occurs, there's folds in the membrane material that's allowing for internalization of those membrane compartments in a process called endocytosis that is internalizing adhesion receptors. So that this compression is really mediating internalization of some of the material that's at that cell boundary, and more work is needed to be able to test that. That's an idea that's been out there, but now we can test the force sensitivities of that endocytosis. If you inhibit endocytosis, you don't get any ratcheting behavior at all. Everything is reversible. Okay. Okay. So what we think is happening is that now there's this ratchet behavior that is a way of buffering the junctions to fluctuations in amplitude, but also duration of signals. If things are too short or the amplitude is too, it won't respond in an irreversible manner. But if you have a strong enough and sustained enough signal, it's going to allow you to permanently change your material properties of that junction in a way that allows you to drive morphogenetic change. So I hope these two stories give you two different scales of this idea of adaptation in epithelial tissue. One is at the junction scale of really thinking about force activated mechanics that's happening within junctions, and at the larger scale of how you can have the tissue architecture control the active stresses. As I was talking to Christoph and Daniel earlier today, what we really would love to do is take some of this information about junction length changes to test the idea of ratcheting behavior in epithelial tissue, to be able to track junction length changes over time to get statistical information of if that's really what's happening. And so that's something that's one area that I'd be really excited to do. And so with that, I'd just like to summarize all the work you heard about today was done by some very talented people in my group. Kate Kavanaugh, John Devaney, and Bob Harmon did all the experiments. Kate was primarily responsible for the later half, and John and Bob were in the first half. And then some really excellent theory collaborators, Sheila Ditcha-Banerjee, who's now at Carnegie Mellon, and Lisa Manning at Syracuse, and Mike and Daniel, who were their students. And thank you for your attention.
Thank you very much for a fascinating talk. Our custom is to allow graduate students or undergraduate students to ask questions before everybody else jumps in.
Yeah.
So are there questions from students? Yeah, Julie.
Hi, I wanted to ask a question regarding the first half of experiments. Have you looked at targeting YAP-TAZ inhibitors at all, since CDK1 is known to interact with the Hippo pathway?
Yeah, that's a great question. Yeah, no, we have not in that particular experiment, but it's something that we're really interested in. We have some preliminary data that I'd be happy to talk to you about. I mean, it does seem like they're. I mean, it's something we're trying to understand in terms of how these features are affecting cell growth and size regulation. Yeah,
Great,
but we did
thank
not
you.
do it in the context of that experiment.
Okay, awesome.
Other questions from students? Don't be shy. Okay, let's open it up to everybody.
Let me get it started. So I don't want to push ahead here, but so
Okay,
it's a
stop.
very general question about sort of understanding of vertex models. I still have sort of a trouble also when somebody asks me to justify that. So the way I think about it is this: so you have a bunch of squishy rubber particles, right? And depending on their pretension and their shape, you can either, if you push on one, you can actually push it beyond the past the other guys, or you can't. Now that sort of implies some kind of dominance of this whole shape story and the springiness of the circumference and the surface area, as opposed to the interactions and the stickiness between the cells. Is there a good argument to say that that should be dominant? Does this whole elastic energy, if you maybe say all the elastic energy in these deformations is larger than the interaction energy or so, is there some good argument for that?
Yeah, so I actually think that there's a self-propelled Voronoi model, which is, I think, more similar to that, where you really are thinking about the individual features, like it's sort of more the compressibility in that way, and then you draw the cell boundaries around that, and then but the vertex model, and I think I probably misspoke. That's and Lisa and I think Andrea Liu have shown that those can map onto each other and make similar predictions. But then the vertex model is really a real justification of the sticky interactions between the cells.
Oh, because you include the
You include the adhesive and the line tension.
that goes into the length.
That's right. Yeah. So but the self-propelled Voronoi models don't include that. That is more like a density, dependence.
Okay.
Michael.
Yes, you talked about this widening of the distribution of tensions during the cell cycle. Yeah. Is there anything about the spatial correlations of those of events or forces or something? Because this is just overall distribution over the whole tissue. Or is there a spatial length scale?
No, that's a really good question. I mean, I mean, let me see if I can go back to this. So that's a little bit chaotic. I mean, sorry, I'm like stumbling. I'm gonna guess there are, but I think they need to be looked at carefully. I think you can still see my screen, right? Because like I think there is like a local mechanical environment and the cell cycle regulation. I mean, there is this coupling between the cell cycle and what we think is the cell mechanics, right? That is going to be controlling the remodeling and the active stresses. And I guess what we don't, this is another thing. We don't really know how, we're trying to do these experiments now of taking cells that are non-proliferative and proliferative cells and making mixtures of tissues with those to see how that affects the overall shape distributions too. Because we don't really quite understand how the spatial distribution of the active stresses affect the remodeling. If it's very local or global, that's one thing. But then we also, I would guess that if the mechanics are, if there's a floppier region of the tissue, that's going to affect the rate of cell cycle, progression, and then that's going to manifest itself in some kind of correlation length. I mean, I'm going to guess there is. We haven't seen that. I think we need different experiments to be able to see it that we haven't seen. I mean, just looking at the movie, you think that there are differences in correlations.
Brent, did you have a question?
Hi, Margaret. Thanks for a great talk. So
Hi.
So the thing at the end where you had, I think, the strain gated tension remodeling. I don't know if I quite understand that, and I don't understand how it then talks to ecoherent recycling. Like, what's the mechanical picture that I'm missing?
I tried to gloss over that because we don't really know. Okay, so so the observation, what we know right now is if you inhibit dynamin, which is an endocytosis factor, you do not get ratcheting. So it's reversible at all time scales. That's one thing we know. And we also know that when there's contraction occurring, you see endocytosis of E-cadherin stuff. And so at the subcellular scale, and that's been observed by other people as well, is that they think that endocytosis is driving adhesion recycling. So what we don't know is what the link is between compression and that endocytosis. Like, where is that compressive stress? So there's been models of that if you relieve compressive stress on, say, membranes or the actin, that's going to allow bending and to increase it. That there is some ideas out there that membrane tension affects endocytosis rates. And I think that would be the one I would favor. I don't have any data for that. I mean, I don't personally, but that's been an idea in the literature.
So I mean, if I can, so then there's kind of like what you think is when you unload the piece of material between the two junctions, you're getting endocytosis of membrane, and that's going to bring in a lot of stuff. So it may not just be the ecoherent, right? Is that?
Oh,
so,
you're saying like a row, it could be row two. Yeah. And so the thing that we really, really don't understand is why we think the active row at the junction should be going up at the end of this. Like, what's controlling the amount of row at the tension at the junction that's allowing it to modulate its amount of actomyosin contractility. But in terms of the endocytosis rate, I would say that the endocytosis rate is inversely proportional to the amount of tension on the junction. So that it goes up and down depending on how much tension is on the junctional, what the junctional tension is. And I think there's actually quite a bit of evidence for that.
I think like Xavier Trepat has something. Like,
exactly.
right.
Yeah.
Right.
Yeah,
but I guess more of what I was saying is I think this could be bringing in a bunch of receptors and other things and not just ECAD. Like, if the membrane's coming in, it's pulling in.
That's right.
Many.
Yeah. It's not just ECAD. It's not just ECAD-herin. That's correct. Yeah.
Okay. Thank you.
Yeah.
Calvin.
Yeah, I wanted to ask a question about the decline of cellular division rate with crowding.
Yeah.
Can I think about this as is there internal stresses that are just intrinsic to the cell cycle, but there are also external stresses due to the nearest neighbors? And so I'm not sure if this is a good way of thinking about it, but have you tried, after cellular division has subsided, to take part of that tissue and move it to a different environment where it may have fewer nearest neighbors, and see if it starts by dividing?
Okay. Yeah. Okay, cool. All right. So okay, so there's two components of this. There's like cell cycle regulation, and there's also cell proliferation, which was the volume. Okay, so that's if you take the tissue and it's crowded, and then you take it and put it into a low density environment, the cell cycle will start cycling again. So the cell cycle regulation is most certainly determined by its mechanical environment. Okay, that we definitely know. And I think Michael might have asked this question, and there should be data on this of showing I'd be surprised if I haven't seen data where you stretch the whole tissue. You could also do this by just putting the whole tissue on a stretchable substrate. get to the point where it's crowded and then stretch it, and then the proliferation rate should increase again. Okay.
All right, thank you.
That answers why. I think you had another question though. Did
Yeah,
you
I
have
had another question.
What was
and
the other
that's
question?
that's about the temporal response of putting the light on the junctions. And
Oh, okay.
so the time if you show one of those slides, I'm looking at time constants of seconds, it looks like to me. Is that intrinsic time constants, or is these time constants dependent on the amount of tissue that you're exposing to the light and measuring the response?
Yeah, these time constants are intrinsic to the molecular affinities under light and dark. There's no cooperative effects that we see. And so these constants are very consistent with what we know in terms of changes in the light and dark affinities.
All right, thank you.
Cool.
Can I ask a sort of what might be a trivial question? I don't know. You have all these cells that are changing shape. You're looking at the top surface. How much do you really know about what's going on? I mean, these things are, the aspect ratio is pretty large, right? They're fairly thick compared to their lateral dimensions.
These particular cells are actually kind of thin. They're only like 5 microns in thickness. But I think you're getting somewhere important. So, but that's, yeah. Like, so, what about the rest of the cell, or?
Yeah, well, I mean, how do you know, like, if you measure the shapes on the top surface, that they're the same even?
Yeah, no, it's a great, it's not trivial at all. It's actually a big criticism of this approach. Okay, so we know that there are some, so in the first project, we know that there are some effects of the basal surface because we did some perturbations to them. But they aren't affecting the cell height at all. And so the shapes are different on the bottom and the top. So what's the question? I think actually I'm actually now shocked. Like, so what the question would be, is the shape parameter on the basal surface a better or worse predictor than that on the apical surface?
Right, and how statistically?
Yeah, and I'm actually shocked that I've never been asked that before, because it's a really good question.
You're looking at the ring, right? You're looking at
yeah, no, it's a great question. What were you saying, Christoph?
You're looking at cadherin labeling, so that's the ring that really binds them together. So
it is,
so
but like we don't have. I mean, yeah, like that would really be an important thing of whether the part that's interacting with the extracellular matrix is how well it's correlated to all these other metrics that we're doing on the apical surface. I can tell you that so a lot of people use this cell type in this field, and we make our tissues a little bit different, so they're more like epithelia. A lot of people make small colonies of them and put them on rigid surfaces, and when that happens, they're a lot more driven by focal adhesions. There's a lot of more motility and neighbor exchanges between them, and ours seems to be more tissue-like. But that's not a good answer to your question. Somebody should just do it. Yeah, no, that's a great question.
Is it possible to image
Yeah, no, we're doing it for other projects in the lab. Like, I'm really currently, and it's probably a horrible project to be pursuing, but like, I'm really curious about like what determines the height of the cells. Like, why do the cells grow to be 5? I mean, I don't understand it. Like, it doesn't make any sense to me. So I have a student, like there are students that are measuring heights of cells all the time. It's not technically, it's actually easier to image the basal surface. Yeah, there's no reason not to be.
It's also a big question and controversy in Drosophila and amnioserosa.
Yeah, no,
so
it's
still an open question.
Yeah, I mean, so with the second project, Kate, one of Kate's thesis members, Edmund Rowe, who some of you know, was obsessed with the idea that the focal adhesions on the bottom were going to be affecting her interpretation of her data, and like, so she did a lot of experiments and showed that there wasn't any mechanical coupling really going on with that were affecting her experiments, but with the first project, that's not true. So we should just do it.
I had a question about the arrest movie. That was really
Yeah.
impressive. That when you turn off the cell cycle, everything stops.
Yeah.
So I guess it looks like from the division events, they don't look strong enough to drive the whole mechanics that you saw before. So can you look separately at the fluctuations of the cells, and do they also stop? So is there another effect?
So I mean, I guess this is going to be hard to do. So fluctuations, what do you mean? Like the
shape and area fluctuations. So they're always wiggling around, like with a minute or so.
What in CDK? Like, look at this. I'm playing the. I don't know if you can see this. There's no fluctuation. Fluctuations just stop. Like
yeah, after the stopping.
Yeah,
but before the stopping, they were dividing and fluctuating, right?
They were, but not all the divisions were doing that. Like so yeah, that's something that's weird, because like the mitotic cells, the cells that you notice undergoing divisions, are only a small fraction of ones in
that's
G2,
what I mean.
right?
That's what I mean.
Yeah.
So it's somebody else helping to drive this whole
That's
thing.
right. That's right. We don't think it's just the mitotic cells. We think it's all the ones in the other parts of the cell cycle too.
That was my impression.
Yeah. But I mean, Christoph, the thing is, like, you use like a myosin inhibitor or any FAC inhibitor, any of these, the motion doesn't stop this dramatically at all. It's really crazy. I've never seen. Yeah.
It's interesting. Yeah. Josh, we're running quite a lot of time here, right? I don't know.
Yeah,
we
sorry.
can talk later, but of course.
Yeah. So we have time maybe for one more question, or perhaps at this point we should sort of officially bring the colloquium to a close so that people who need to leave don't feel bad about it, and we can keep the session open for a little while for anybody who wants to hang around and talk a little bit more. I think Professor Gardel's next meeting is at 5. Is that right?
There was supposed to be one at 4:30 with Michael, but I'm the last one. So Michael, you can just start whenever we're done here, and then either I chat with Margaret if she has still time after that, or some other day. So there's definitely many questions I have.
So thanks everybody for joining.
Well, thank you.
Stick around for a minute or two.
Thank you.
If you want to ask a question.
Great talk.
So before I let you go to Michael, I did have one more question. That's a kind of, I don't know whether well, I'll just ask it and get your reaction. The question is whether these vertex models or Voronoi models, do you think of them as mechanistic models or just as phenomenological models? You know, how do you tell whether they fit the data? You know.
Oh God, this is a hard question.
Yeah, it's my struggle too.
I mean, you can make a model, you can put in dynamics, you can watch what happens, and you can say this movie looks like a real movie. Okay, so I think like the stuff with Sheila, right? Like that, where we're trying to where we start getting rules of like how we can actually test it. I think with the optogenetic ways you can actually test it. So, but whether that's mechanistic or not, but the models themselves are not based on sort of representing microscopic. Oh, that question. So I know I struggled with this for a while, and I think I just folded. I think
phenomenological.
I know.
They are. Exactly. The only hope is that they're simple enough to make to sort of.
It's surprising to me how well they work. It's actually really surprising to me. Like I fought against it for so long, and I think I finally, it is surprising to me. Mm-hmm. But but this whole issue of dynamics is really something that they don't do well at all. Which is why I'm kind of interested in that aspect of it, right? Like, anyways,
so
I didn't mean to interrupt. I would say phenomenological but useful.
Exactly. It could point to some deeper truth because of their simplicity. Right?
So I'm going to stop the recording now.
Okay. Oh, that was recorded.
Oh.
No.